Cellular transport. Learn the role that the cell membrane plays when transporting material in and out of the cell. When we talk about diffusion or osmosis regulation, there's a few words to know. This must be from like physical science or even maybe from an earth science class you might have had. A solute, that's what's getting dissolved. Okay, that's like the lemonade powder that gets dissolved inside of the water. The water is the solvent. That's what's doing all the dissolving. Together, they form a solution, a relatively uniform mixture of these two substances. Now, how much solute I put in gives me a concentration. So I can make the lemonade more tart, if I like, by putting more powder in. I can increase the concentration of solute in the solution. Usually this is abbreviated by brackets. A concentration amount will oftentimes have brackets around it. That's how you know they're talking about the concentration of something and not a numerical measurement of something else. A concentration gradient is the difference in the concentration of substances from one location to another. And like I said, passive transport kind of moves from that high concentration to low concentration. It's flowing from one location to another, moving down a concentration gradient. This movement down a concentration gradient will happen until things reach an equilibrium or a happy state. Equilibrium is when equal concentration is equal on both sides. So it kind of levels out, if you will. And when this is happening across the membrane, the molecules moving across the membrane are going to simply diffuse until they reach a point where the concentration inside of the cell is the same as outside of the cell. Now, molecules will move down a concentration gradient from high concentration to a low concentration. So, for example, here I have a membrane. Oops. And this is my barrier right here. I have a lot of salts on this side and very few salts on this side. So things are going to move in this direction. It wants to level it out. It wants to even the playing field. So over time, they're going to move in this direction. Now over here, you can see this is my membrane, and I have relatively the same concentration of particles over on this side as I do on this side. So this, we can say, has reached equilibrium. The parts have kind of leveled out. Now, facilitated diffusion is usually done through a transport protein that helps help along, the diffusion of molecules that normally couldn't pass through the cell membrane. So transportation proteins, they act as like a channel or a carrier. I like to think about it as a very particular shaped slide. And that molecules will move down a concentration gradient from high to low. Large molecules, like the big 6-ring sugar glucose, and polar molecules like calcium are just two examples of molecules that move through a protein channel. So here I have my same barrier again. I have a high concentration over here and a low concentration of particles over here, but there's a channel. There's a protein channel, which means only certain size things or certain shaped things are going to be able to move. And because they move technically still passively, they're having to fit in this tube to get to the other side. And they'll continue to do that until they reach equilibrium. When you have something that is equal on either side, like here's our membrane and here's our channel, nothing's going to be moving. It's not moving in this direction or in this direction at all because it's equal on both sides. And so that's really not going to have anything to do with the channel. When you were little, did you ever play with those baby toys where it was a ball, and on the outside of the ball there were different shapes like a star and a circle and a square, and the baby had to find the shape and find the hole that it would fit into? It's kind of like that. what's going through this channel is specific in shape. It's facilitated. It's helped along through a special shaped passage, if you will. It still doesn't require any energy. Our last type of passive transport is osmosis. And osmosis is the diffusion of water. We call it simple diffusion because we're just talking about water here. Water molecules move down a concentration gradient from high concentration to an area of low concentration until we reach equilibrium. High water concentration means low solute concentration. So if you're looking at this part over here, if this is my membrane, over here it is very, we'll just say salty. Let's just say these are salts. So there's a high concentration of salts over here and a low concentration of salts. So if we're talking about the water where the salts are sitting in, it's going to move in this direction because it wants to dilute this to make it the same concentration as it is over here. And what ultimately happens with, over time, is this membrane, which is allowing water to move through, but not the salts, will increase in volume. We'll get more water on this side because it wants to move to evenly distribute these particles, these salt particles, away from each other so that the concentration is the same on this side as it is on this side. Where this typically happens that we care about in studying biology is in blood cells. Blood cells are hypertonic, where water is lower than the cell's cytoplasm concentration. And so we see this net movement of water out of the cell and the cell shrivels up. Hypertonic. Or cells can be in a hypotonic, where water is at a higher concentration than the cell cytoplasm. So the net movement of water goes into the cell and the cell swells. Hypotonic. Then isotonic solutions, those are solutions that are identical to the concentration of the cell. So the cytoplasm doesn't move and the cell stays the same. So these are usually how we see the scenarios. Here I have my cell and its concentration of water inside the cell is very, very high and its concentration outside the cell is very, very low. So the water is going to want to move out of the cell to the area where water concentration is lower. We call that hypertonic, and then the cell shrivels. In hypotonic, the cell has a very small concentration of water, and outside is a very large concentration of water. So this larger concentration wants to equal this out, so it's going to move inside of the cell, making the cell swell from this size to a larger size. See, what it's doing is it's wanting to osmoregulate, keep this cell, have the same concentration of water inside versus outside. In isotonic, you can see that the concentration is the same, both in and out of the cell. So the cell is going to stay the same. This is easier if we practice it. Here is my cell. It has 20% sodium chloride and 80% water. And my environment has 10% sodium chloride and 90% water. What do you think is going to happen? Pause and practice. The water is going to want to move inside of the cell because it's lower water concentration on the inside, and this is a higher concentration. So the cell is hypotonic and it will swell. Let's try another example. Here I have a cell. It's 5% sodium chloride and 95% water. And on the outside, its environment, it's 15% sodium chloride and 85% water. What do you think will happen? The cell's going to shrivel because the solution on the outside has a lesser concentration of water. It's higher concentration of water on the inside. So the cell is going to shrivel up. It is hypertonic. And water is going to move from inside of the cell to outside of the cell. How about this scenario? We have a 10% sodium chloride solution and a 90% water solution. 90% water solution inside the cell. And the environment has a 10% salt concentration on the outside and a 90% water concentration on the outside. What's going to happen to this cell? This cell is going to stay isotonic, which means it's going to stay the same. Now, water is still moving in and out of the cell. We just don't notice any difference in the cell size because it's in an isotonic solution. It's maintaining its shape.